All right, let's dive right into this fascinating breakdown of the integrated stress response. This is basically an incredibly elegant emergency protocol that literally keeps our cells alive. If you've ever wondered how your body handles microscopic crises at the cellular level, well, you are definitely in for a treat today. We're going to walk through this entire explainer to see how a single cell acts like a highly sophisticated supercomputer when things go terribly wrong. So, picture a cell just under absolute siege from multiple fronts. I mean, it could be starving, it could be infected by a virus, or its internal machinery could just be failing. The crucial puzzle here is understanding how it orchestrates a unified defense to actually survive. It can't just panic, right? It needs a highly calculated master blueprint to deal with these vastly different threats. And that blueprint is exactly what the integrated stress response is.
Okay, moving to Section 1, the 4 stress sensors, which serve as our front lines of defense.
To start our journey, let's check out these front lines. Think of this setup like a giant funnel. We've got 4 distinct crises. If the cell starts piling up misfolded proteins, the PERK sensor fires. If there's a viral infection, usually flagged by double-stranded RNA, the PKR sensor goes off. Starving for amino acids, that trips GCN2. Or what if there's a sudden lack of heme or some sort of mitochondrial crisis? The HRI sensor detects that. So whether it's a virus or straight-up starvation, you have 4 completely different kinases acting as these highly specialized threat detectors guarding the cell's perimeter.
But here's the real genius of the cellular design. No matter which of those 4 sensors is tripped, they all funnel their distress signals into one single universal alarm switch known as EIF2 alpha. They pull this off through a biochemical process called phosphorylation, by basically translating these totally different languages of stress into one common biological signal. Phosphorylated EIF2 alpha, the cell guarantees a massive, unified response without needing 4 completely separate defense grids.
Let's jump into Section 2, Translation Halt, focusing on the EIF2 alpha hub. Now that this universal alarm is blaring, what actually happens mechanically inside the cell? Well, you kind of have to think of your cell as a massive protein manufacturing factory. Usually there's an enzyme called EIF2B that acts as a recycler. It's constantly regenerating the fuel needed to start building new proteins. But when our universal alarm is present, that phosphorylated EIF2 alpha, it actually attacks and blocks that recycler. The recycling process plummets, the complex needed to start translation just fails to form, and boom, global protein manufacturing drops off an absolute cliff. By simply slapping a phosphate group onto EIF2 alpha, the cell ingeniously grinds the entire factory floor to a halt. The big takeaway here, slowing down that factory floor is literally the ultimate biological triage strategy. Think about it. If you're starving or actively fighting off a hostile virus, the absolute worst thing you could possibly do is waste your precious limited energy building normal, everyday proteins. You've got to shut down non-essential operations immediately to conserve resources for the fight ahead.
All right, Section 3, the ATF4 paradox. This is a brilliant twist in biology.
Just when you think you've got this whole system figured out, we hit a massive biological plot twist. The sheer irony here is incredible. On the one hand, global protein synthesis is completely off. The factory is totally dark. But on the other hand, the production of one specific protein, a crisis commander called ATF4, is actually turned on, and its production skyrockets. Now, how does it bypass a total lockdown? Well, ATF4 messenger RNA contains these special biological bypass codes called upstream open reading frames. They basically act like an all-access VIP pass, allowing ATF4 to be translated exclusively while everything else is bolted shut.
So what does our VIP commander do once it's deployed? ATF4 completely rewires the cell. It shifts all priorities from normal operations to a highly specialized emergency survival program. It heads straight to the nucleus to activate genes for things like amino acid metabolism, antioxidant defense, and autophagy, which, if you remember, is essentially the cell recycling its own damaged parts for fuel. ATF4 is the master architect of metabolic reprogramming, and it gives the cell a real fighting chance to adapt and recover from the crisis.
Which brings us to Section 4, the brake release, looking at the GAD34 feedback loop. Now, obviously, living in a constant state of total panic would be fatal. You can't leave the emergency brake pulled forever. The cell needs a way to turn the alarm off. It plays out in a beautifully choreographed negative feedback loop. At minute 0, our crisis commander ATF4 hits its peak. But ATF4 knows its job is strictly temporary. So around minute 30, it actually triggers the creation of a brand new protein called GAD34.
By hour 1, GAD34 acts almost like a biological tow truck. It brings in an enzyme called PP1 to physically rip that alarm-triggering phosphate group right off of EIF2 alpha. By hour 3, the alarm is completely silenced, and the factory goes back to normal. This brilliant loop shows how GAD34 safely returns the cell to normal operations once the danger is actually gone.
Moving to Section 5, Mitochondria and Heme, looking at supply and demand. Let's zoom out from these microscopic proteins for a second and connect this to something tangible we all know, our blood and oxygen supply. Think about the red blood cells in your body. To make functional hemoglobin, the cell needs two things in perfect balance: heme, which carries the oxygen, and globin proteins. If the heme supply suddenly drops, maybe you have an iron deficiency, but the factory floor keeps blindly churning out globin proteins, you get a catastrophic backup. The unmatched globin chains clump together and literally destroy the cell from the inside out. This is exactly why the HRI sensor from earlier exists. It detects the drop in heme and immediately triggers the stress response to shut down globin translation. It perfectly balances cellular supply and demand. But actually, it goes even deeper. Modern research has mapped out this stunning relay race, showing how a distress signal physically travels from inside the mitochondria all the way out to that HRI sensor. When the mitochondria experience stress, a protein called OMA1 gets activated. It cuts up another protein, DELE1, which then literally travels out of the mitochondria into the main body of the cell to find and activate the HRI sensor. It's this breathtaking physical chain of command that proves the mitochondria aren't just dumb powerhouses. They're active, dynamic sentinels, constantly monitoring cellular health. And finally, Section 6, Cell Fate Decision, to live or to die. All of these sensors, loops, and cascading signals ultimately lead to one incredibly dramatic conclusion. This entire pathway forces a final monumental decision for the cell: to live or to die. Following our crisis commander ATF4, another protein steps onto the stage called CHOP. For a long time, scientists thought CHOP was just a grim reaper, a guaranteed death switch. But as we track the timing, it's far more nuanced. In the short term, CHOP actually helps regulate the stress response to promote recovery. But if the stress is just too intense or lasts too long, CHOP crosses a threshold and pushes the cell toward apoptosis, which is basically programmed cellular suicide, just to protect the rest of the organism. Cell fate is beautifully mathematically calculated based strictly on the intensity and duration of the stress.
Because this system is so fundamental, scientists have actually developed incredible ways to interact with it. Researchers use chemical cheat codes like ISRIB and salubrinol to artificially manipulate this life-and-death system in the lab. ISRIB, for example, stabilizes the cellular recycling machinery so that even if the alarm is blaring, the factory can partially ignore it and keep making proteins. Salubrinol does the exact opposite. It blocks the brake release, keeping the alarm blaring artificially. These tools are completely unlocking entirely new understandings of diseases related to cellular stress.
When you pull all these complex threads together, you arrive at this profoundly elegant biological narrative. Simple building blocks like heme are way more than just structural pieces of a puzzle. Heme is a master signaling molecule. It holds the ultimate authority over the entire protein manufacturing grid, bridging the gap between how much energy a cell has and whether it decides it's safe enough to survive and grow.
It really just makes you pause and appreciate the invisible intelligence keeping us alive every single second, doesn't it? I hope this breakdown leaves you marveling at the sheer genius of the integrated stress response. I mean, if our cells possess such a brilliantly calculated emergency master plan to triage, adapt, and recover, what other hidden biological codes are waiting to be uncovered? Keep questioning, keep learning, and I'll catch you on the next explainer.